Destroying harvests for millennia, the stem rust pathogen of wheat is a sophisticated organism that continues to this day to threaten the security of global wheat production. The emergence of the UG99 group of virulent stem rust races underscores the need to better understand the mechanisms of resistance in wheat and to develop new varieties resistant to current races of stem rust. But the emergence and rapid spread of new forms of virulence also prompts more fundamental questions. How do new virulent races arise in the first place? And what factors contribute to the rapid and ongoing evolution of this historic pathogen? Stem rust of wheat is named for the powdery, reddish-brown, rusty-colored blisters or pustules that develop on the stems and leaves of susceptible wheat lines grown under conditions conducive to infection. This telltale reddish-brown powder found on infected wheat plants are spores of the fungus Coccinia graminis, the organism that causes stem rust. A single pustule can produce tens of thousands of these spores, called uridinospores, with each one capable of infecting a wheat plant and producing yet another pustule. Carried by the wind, perhaps to a neighboring plant, perhaps to another wheat field hundreds or even thousands of miles away, a microscopic uridinospore can land, germinate, and cause new infection. Once inside the host tissue, the pathogen extracts nutrients from the wheat plant's cells, grows, and eventually produces a new generation of uridinospores in such abundance that the plant's tissue erupts, releasing the new spores into the air to repeat the infection process elsewhere. On and on the cycle goes. With roughly 2 weeks from infection to sporulation, this cycle of uridinospore multiplication can rapidly produce epidemic levels of disease. A severe stem rust infection in the field is a dramatic and devastating sight, but it is not the whole story. An important point about the uridinospore cycle of infection is that it is a clonal method of spore production. As the growing season comes to an end and wheat plants begin to set seed, senesce, and die, the stem rust pathogen does something remarkable. Instead of producing more uridinospores, it begins to produce black overwintering spores called teleospores. At which point it can germinate and produce through meiosis haploid spores called basidiospores. Incredibly, these basidiospores infect not wheat, but certain species of the completely unrelated woody perennial plant genus Berberis, or barberry. Transported by the wind to barberry plants near wheat fields, basidiospores can germinate, infect, and create blisters called spermagonia on the upper surface of barberry leaves. These blisters produce reproductive cells called spermatia, able to fertilize receptive cells called hyphae inside compatible neighboring spermagonia. In this step of fertilization, entirely novel genetic combinations are achieved. Once fertilized, the hyphae grow down through the barberry leaf, where they eventually produce stunning reddish pustules on the underside of the leaf, marked by numerous peach or orange-colored tube-like structures called eecia. When exposed to moisture, these eecia, quite visible to the naked eye, forcefully eject the next spore form of stem rust, eesiospores. Carrying entirely new combinations of genetic material due to the cycle of sexual recombination on the alternate host, barberry, these microscopic eesiospores can once again directly infect wheat, producing the brownish-red uridinospores that are the hallmark of this disease.